Let's review. Both animals and fungi have to ingest food as a source of carbon, either by digesting or decomposing food. Neither fungi nor animals can photosynthesize. As was mentioned earlier, these processes are happening at too small of a scale within cells to be able to be directly observed. To measure animal processes, scientists must do studies where we observe organisms or the air around the organisms and take measurements. In this experiment, scientists measured the carbon dioxide released by rats multiple times over a 2-hour period. They used a device called a respirometer to measure the concentration of carbon dioxide gas in the tube released by rats. Over time, you can observe an increase of carbon dioxide gas concentration in the air in this chamber. Looking at the y-axis of the graph, this increase is quite substantial. There's not a lot of carbon dioxide in the air, only about 0.04%. So the carbon dioxide released or taken up by organisms can be quite important at the scale of organisms and ecosystems. The scientists also measured the oxygen taken in by rats in cellular respiration using the respirometer. There is a decrease in the concentration of oxygen gas in the chamber over 2 hours. Again, take a look at the y-axis of this graph. Compared to the total amount of oxygen in the chamber, the change in oxygen isn't very substantial. This is because there's a lot of oxygen in the air, about 21%. So the oxygen released or taken up by organisms is less important at the scale of organisms and ecosystems. In the same experiment, the scientist weighed the rats multiple times over the course of 2 hours using a very sensitive scale to calculate the change in mass of an individual rat. Rats were not fed during the experiment, so any changes in mass can be attributed to cellular respiration. As you can see, the scientists observed a decrease in mass over time. Again, note the scale on the y-axis of this graph. The changes in mass are very small, but definitely observable. Now, imagine an organism in a closed system, in this case, inside this sealed box. No air can come in or out of the box, but sunlight can get through. If you were to monitor the change in oxygen over the course of several hours and noticed an increase in oxygen, what biological process could have caused this? To answer that question, let's place it in the context of the patterns and processes we've been talking about. The pattern we are observing in this case is the change in oxygen concentration in the box, and we're seeing an increase in that oxygen concentration. Since only organisms that photosynthesized could have caused an increase in oxygen concentration, we can conclude that whatever is inside the box is undergoing photosynthesis. Now, imagine an organism in a closed system, in this case, inside this black box in which no sunlight can penetrate. If you were to monitor the change in carbon dioxide over the course of several hours and notice an increase, what biological process could have caused this? Again, let's place this question in the context of the patterns and processes we've been talking about. The pattern we are observing in this case is the change in carbon dioxide concentration. And in this case, we're seeing an increase in carbon dioxide concentration. We would expect to see this increase if the organism inside were undergoing cellular respiration, since carbon dioxide is a byproduct of that process. So we can conclude that whatever is inside the box is undergoing cellular respiration.